Welcome back to the Learning Studio. Today marks the start of a brand new section in our Biological Sciences series, Genetics and Genomics. Over the next few episodes, we'll explore how traits are passed down, how populations evolve, and how modern genetics is reshaping medicine and biotechnology. If you'd like to catch up on earlier topics from molecular and cellular biology, the full playlist is linked below in the description. Here's something to think about. Why do you have your mother's eyes but your father's height? And why do some traits, like blood type, seem to skip generations? The answers lie in the rules of inheritance, first uncovered by Gregor Mendel, and later expanded into what we now call Mendelian and Non-Mendelian inheritance. Mendelian inheritance, the classical rules. Gregor Mendel, the father of genetics, studied pea plants in the 1800s and discovered patterns that still form the foundation of genetics today. His work gave rise to Mendel's laws. Law of segregation. Each individual has two alleles for every trait, and these alleles separate during gamete formation. Law of independent assortment. Alleles of different genes are inherited independently of each other. Law of dominance. Some alleles are dominant, masking the effect of recessive alleles. Examples: eye color inheritance, pea plant traits, tall versus short, green versus yellow peas. Human conditions like cystic fibrosis, recessive, or Huntington's disease, dominant. Non-Mendelian inheritance beyond the basics. But not all traits follow Mendel's simple rules. Modern genetics has uncovered non-Mendelian patterns, including incomplete dominance. Neither allele is fully dominant, e.g., red-white flowers as pink flowers. Codominance. Both alleles are expressed equally, e.g., blood group AB. Multiple alleles. More than two alleles exist for a trait, e.g., ABO blood groups. Polygenic inheritance. Traits controlled by many genes, e.g., human height or skin color. Mitochondrial inheritance. Traits passed through mitochondrial DNA, inherited only from the mother. Epigenetics. Environmental factors switch genes on or off without changing DNA sequence. Case studies and examples. Case study 1, sickle cell anemia, shows incomplete dominance. Heterozygotes have partial resistance to malaria. Case study 2, blood types demonstrate both multiple alleles and codominance. Case study 3, height in humans, a polygenic trait influenced by both genetics and environment. Why it matters in genetics and genomics. Understanding inheritance is the gateway to modern biology. It explains family traits, helps doctors predict genetic disorders, and even informs agriculture and breeding programs. Without Mendelian and non-Mendelian principles, we couldn't make sense of DNA sequencing, gene editing, or personalized medicine, past, present, and future relevance. In the past, Mendel's peas laid the foundation of genetics. In the present, genetic counseling helps families understand inherited risks. In the future, CRISPR and advanced genomics may allow us to correct faulty genes and reshape inheritance itself. Why watch this playlist? This playlist covers the 30 most important topics in biological sciences, explained step by step with examples, case studies, and real-world applications. Whether you're a student preparing for exams, a teacher searching for resources, or a curious mind exploring science, this series is designed to help you build a solid foundation in biology. So that's our introduction to Mendelian and non-Mendelian inheritance, the rules, the exceptions, and the fascinating ways traits pass through generations. In our next lecture, we'll continue the genetics and genomics section with population genetics and evolution, diving into how genes behave not just in families, but in entire populations over time. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss the exciting journey through genetics and beyond. Because once you understand inheritance, you start to see the blueprint of life unfold.